All right, so today we're going to be talking about brain and behavior. That's part of chapter 3. And we're going to be doing introduction of neuroscience. Neuroscience is a really important part of psychology, and it's going to help us understand the biological foundations of the brain and cognition. So why does this class include neuroscience? Well, neuroscience is the study of how nerves and cells, particularly neurons, send and receive information throughout the brain, body, and spinal cord. And it's important because the way that the biological makeup of our brain will influence how we perceive things in the world, how we take in information, how we behave, and how we act within after getting that information, what kind of things we do. So I'm going to be talking about that and how that works. So one question that could be going through your mind is, what is neuroscience exactly? So neuroscience is the study of how nerves and cells send and receive information from the brain, body, and spinal cord. And like I mentioned, they include neurons, which are specialized cells found in a nervous system. So how do we know anything about the world around us? How do we get that information? So the reason why we get that information is because these neurons, which are specialized cells, make up our nervous system. And understanding how those neurons work is the function to how we can decipher how we perceive the world around us. And one way to start this conversation would be understanding that there's two types of processing within the brain. So there's bottom up and there's also top down. Bottom up means that we receive information from our senses. And when we gather that information, we try to make sense of these pieces and determine what is happening. And then from a top-down approach, it also influences our lower regions of our brain. And we're able to behave and create action based on past experience and knowledge that we have. So remember, bottom up and top down. Another thing that would be important to talk about is the cerebral cortex. And the cerebral cortex is made up of 6 layers of cells on the surface of the brain, and that's called gray matter. That's a technical term. And this supports all thought, emotion, and complex processes. From an evolutionary standpoint, it's very recent when it comes to with the human evolution, and it's a very sophisticated system. I want to bring up this quote by Francis Crick, and Francis Crick had mentioned, "You, your joys, your sorrows, your memories, and your ambitions, your sense of personal identity and free will are in fact no more than the behavior of a vast assembly of nerve cells and their associated molecules." So I'm just wondering what you think about this quote. Let's give you a moment kind of to process it. I think this quote is interesting because it challenges our perception of free will and then also consciousness. Do we really have choices in the things that we do if our brain structure is the way it is? And is the way that our brain structure is made up influence our decisions on a daily basis and for our whole entire lives? So you'll see linked in the slide is a TED Talk by Jill Bolte Taylor. And Jill Bolte Taylor had studied her own stroke as it happened and became an advocate for brain recovery. And within the video, you'll see that her experience emphasizes the importance of the different brain hemispheres and shaping consciousness and identity. So I just want you to make sure that you watch the video. It's very interesting about her experience, how she reflects an experience, and how that informs our understanding of the different hemispheres of the brain. So let's talk about the neuronal structure. So the different parts of the neuron include the dendrites, cell body, axon, myelin sheath, and axon terminals. And remember that the neurons are the building blocks of the brain. It consists of these structures, and this is how we're able to send and receive information between brain cells. So a little bit about the neuron signaling is that neurons send signals both within and between the neurons. So the signals in neurons are electrical. The signals between neurons are chemical. So as you can see right here, there's electrical impulse that travels down the axon, and that's, remember, it's electrical. And then there's chemicals that will go between each neuron. One thing that would be important to talk about is the glial cells. And the glial cells insulate, support, and nourish neurons and modulate neural functioning. They're responsible for vacuuming up neuronal debris and serve as kind of like a glue between neurons, giving the brain its jelly-like consistency. And it's really important for brain development. So the next thing we're going to talk about is the nervous system. And the nervous system is like a complex set of neurons that extends throughout your body and your brain. And this network includes cells and carries signals from various parts of the body to the brain and allows that communication and response. So the nervous system, like I was mentioning, runs throughout our brain and our body, and it's a collection of nerves, um, and which are made up of neurons that carry signals from the body to the brain. And there's three types of neurons. There is a motor neuron, there is an interneuron, and there's also a sensory neuron. So the motor neuron sends signals to muscles and prompts physical actions. The interneuron is located primarily in the brain and the spinal cord, and it processes information, allowing for complex decisions and responses. And the sensory neuron gathers information from the environment and the body, transmitting it to the brain. Our nervous system can be divided into two parts. There's the peripheral nervous system and the central nervous system. So the central nervous system is made up of the spinal cord and the brain, which I just talked about. And the peripheral nervous system is made up of the somatic nervous system and is made up of the autonomic nervous system. And I'll explain those different parts. So the central nervous system is comprised of, like I mentioned, the brain and the spinal cord, and it helps process information through various channels. The peripheral nervous system consists of nerves outside of the central nervous system. It connects our limbs and our organs to a central hub for processing. The peripheral nervous system can be separated into two different divisions, and that's the somatic nervous system, which controls voluntary movements, and the autonomic nervous system, which controls involuntary functions like heart rate. And just to give you a little bit more detail about that, the nervous system for the autonomic nervous system, it is divided into the sympathetic nervous system or fight or flight response, and the parasympathetic nervous system, which is when it comes to resting, what is our body doing while we're resting, or when we're digesting food and those processes and things like that. And if you're wondering for fight or flight, what that is, that's a sympathetic nervous system response. And it's like a response when we're encountering dangerous or stressful situations and prepares the body for action, increases heart rate, blood flow to the muscles, and then dilates our pupils. And then some more information when I mentioned rest and digest, that's part of the parasympathetic nervous system. And that works to calm the body down after a stressful event, slows down our heart rate, and conserves energy and promotes digestion. So this diagram is really helpful with identifying the different processes when it comes to sympathetic and parasympathetic nervous system. So as you can see here, like I mentioned, the sympathetic nervous system is made up of the brain and the brainstem and the spinal cord. And as you can see here, it connects with all different parts of our body through the spinal cord. So I want you to make sure to watch this video. It talks about the future of brain-computer interfaces, or BCI. Could also be called brain-machine interfaces, BMI. And as we understand more about the nervous system, these technologies like the BCI or BMI are definitely advancing. So you'll learn about how these interfaces allow for direct communication between brain and external devices. And after you watch the video, some things to think about would be, what are some potential future developments of this technology for BCI and BMI? What are the implications when it comes to medicine, technology, or even just our daily lives? The next thing that I'll talk about is the endocrine system. And the endocrine system are a network of glands that produces and releases hormones into the bloodstream to regulate the body's activities. And it works with the central nervous system to synchronize the brain and the body. It has a slower rate of transmission than the nervous system. We have the hypothalamus, we have the thyroid gland, we have the pancreas, the ovary, testes, adrenal glands, parathyroid glands, and pituitary gland. And as you can see here, these all have different jobs when it comes to transmitting hormones. And what are hormones? So hormones are blood-borne chemicals, and they travel throughout our bloodstream, and they enable the brain to regulate the body's activities. So each of these hormones have different jobs for the body. Let's talk about the difference between neural and hormonal mediation of behavior. So when it comes to neural mediation, it can directly trigger behaviors. And there's fewer targets in the body, typically only the cell, typically the only cells that they're not in contact. And then when it comes to hormonal mediation, these rarely trigger behavior, but they change the stimulus threshold necessary for behavior. Many targets with the body can be reached using this mediation. And some of the components of the endocrine system, so I talked about these, I listed them earlier. So what do each of them do? So the pituitary gland is the master endocrine gland, we would say, and it modulates hunger, sexual arousal, and sleep. The adrenal glands use a variety of hormones. One of the most notable ones are adrenaline and cortisol. Maybe those chemicals sound familiar to you. You've heard them before. And some of the things that could be influenced would be the energy levels of our body, increasing our heart rate, and it's central to our stress response. And that's activated by the central nervous system. So there's different classes of hormones. You won't need to know every single kind of hormone, but um, here are some ones to mention. So amino acids and derivatives, like epinephrine, for example, impact adrenal medulla. And peptides and proteins have short and long chains of amino acids. Steroids are synthesized from cholesterol fat and are fat soluble, able to enter cells and bind to receptors in the cytoplasm or nucleus. So some things you might have learned in biology class. For example, all sex hormones are used for sex development and behavior. So just kind of going back to the difference between hormones and behavior and how they interact with each other. So hormones and behavior are interlinked. We have our sensory systems, which help us perceive the world, and those signals go through our central nervous system. And then our output systems, and then that helps modulate our action systems or output systems on what kind of to do next. And that influences our behavior. And throughout this process, hormones are integrated and affect those systems. So the next thing that I want to discuss is the bonding and parental care in the prairie vole. So that's been studied many times, and it helps us create an understanding of oxytocin and vasopressin, which are different chemicals within the brain. And these chemicals influence the bonding and parental behavior of the prairie voles. For when it comes to oxytocin, it has to do with female sexual activity, and that helps form a monogamous pair bond with sexual partner. And so that's kind of the chemical that's released with oxytocin. And then when it comes to vasopressin, it creates a similar pair bonding effect in males and could be related to parental care. So the studies with the prairie vole have helped us understand those chemicals. So some things to understand, I'm going to go back to this diagram that we had looked at earlier, is our relationship between hormones and neurons are bidirectional. So what that means is it kind of goes back and forth. And so our sensory systems, our central nervous system, which remember are made up of neurons, they are continually interacting with our hormones. So it has a bidirectional relationship. And when we're exercising, when we go through stress, that is always influenced by hormones and then also by the neurons and their signals. And some other things to remember, remember that chemicals within our brain and our body will influence our behavior. Some of those notable chemicals that will influence our behavior could be cortisol or testosterone. And those are chemical messengers that help modulate and change our behavior and actions. All right, so the next example that I'm going to show you is with the Japanese quail. Please excuse my writing. My writing is really messy. These are just notes that I took as I was looking at this slide. So behavior can affect the hormone levels of Japanese quails. And one thing they looked at for this study was copulation. And when different quails are paired, there's different effects when it comes to the chemicals in their brains. So when they're paired with each other, a male and a female, it starts production of aromatase. So the more time that a male spends looking at females, the increase of production of aromatase. And so some of those chemicals also involve testosterone. So some of the behaviors that could be influenced by these chemicals would be aggression and reproductive behaviors. And then also exposure to predators will influence this production. So production of testosterone and aromatase might go down if there is more exposure to predators. Some other things that are found were that testosterone increased with more social interactions. And then when there's more stress, there'll be some kind of adaptive response, and these chemicals like testosterone and aromatase would decrease. And this is an example of how chemicals within the brain can influence behavior. So next, let's go back to talking about the cerebral cortex. So let's talk about the brain in its different parts. So there's three large divisions in the brain, the forebrain, the midbrain, and the hindbrain. So as you can see there, our forebrain right here, and then our midbrain right here, and then our hindbrain, and then the spinal cord right here. The other parts of the brain that we should talk about would be the different lobes of the cerebral cortex. So the frontal lobe right here, on the parietal lobe, the occipital lobe, and then the temporal lobe. And I'll talk about the different functions, those different systems in just a bit. So let's just take a moment to look at these different types of brains from different animals, including humans. So just take a look at that. What kind of things do you notice? What kind of differences do you see? We have a human brain right here. Which brains do you think look closest to the human brain? And what does this tell us about the evolution of humans? The other thing that I wanted to note is the graph that's on the right. So the graph tells us, it kind of compares the body weight of the individual or the animal and the brain weight. So as you can see, we talked about research methods and how to read graphs in our last session, that there's a positive correlation between body weight and brain weight. As the body weight goes up, then the brain weight goes up as well. So take a look at right here. This is where humans are. So there is an article that's linked within the slide if you want to learn more about this. It'd be interesting to hear your thoughts about why there's a positive correlation between body weight and brain weight. And so what does this say about mammals? So the next thing we're going to talk about is the neocortex, and that's that outer layer of the brain. And that's when we look at a brain, it's very wrinkled and folded, and it's newest in the terms of evolution. And it supports complex thought, like language, like problem solving and imagination. So that neural cortex is, we also call it like the thinking brain. And we have our emotional brain, which is the limbic system, which we'll talk about, and our survival brain, which is kind of like a reptilian part of our brain, which has to do with more basic things like within our body processes. So this diagram is a somatosensory and primary motor cortices. And I guess the best way to explain it is if we opened up someone's skull and take a surgical steel probe and pass a voltage through, we can activate different neurons in the sensory and motor cortices. And then by doing so, we can map out where each part of the body sends information and where voluntary emotion originates. So as you can see in this diagram, if we did open up the brain and we used a probe to poke at the different parts of the brain, we would know which parts of the brain are associated with the parts of the body and where the signals from the brain are sent. And then when the part of the body, when there's input information, where that signal goes to in the brain. Okay, so let's talk about the association cortex. And what that does, it originates information coming in from senses with existing knowledge and also produce experience of the world. And it's present in every lobe. So as you can see here, there's different parts of the association cortex, and it can be divided up into these different auditory and visual sections of the brain. And locations of major associating areas of the cerebral cortex, as well as the primary and secondary motor and sensory areas, are comprised of the association cortex. And let's talk about how the structures of the brain influence our emotions, our motivation, and our memory. So beneath the cortex is the subcortical regions of the brain. And we would call this as like the older part of the brain when it comes to evolutionary standpoint. It's clusters of neuronal cell bodies called nuclei. And we have our frontal lobe, our parietal lobe, and our occipital lobe, which is made up of the cerebral cortex. But then we have more subcortical regions, which is we could consider the limbic lobe part of that. And I'll talk about that along with some of the other structures. So the limbic system, which I just mentioned, bridges older lower brain regions for body regulation, and it bridges that with newer processes, which are the more complex processes in the cerebral cortex. And it's also described as the emotional brain and plays a role in our smell, learning, memory, and motivation. So the limbic system is part of that, and it's made up of and it's made up of the thalamus, the hippocampus, the amygdala, the hypothalamus, and the basal ganglia. So the hippocampus is a component of the limbic system. So it's paired one on each side of the brain, and some of its functions are for memory, ability to navigate in the environment, and to also think about the future. We have the hypothalamus right here, the amygdala, and the hippocampus. And remember, those are part of the limbic system as well. One thing to look at is the video on spatial memory and taxi drivers in London. That is an important thing to watch because there's studies which are done by MRIs, which we'll talk about in a little bit too. And so the MRIs of London taxi drivers show that extensive navigation or extensive experience being taxi drivers in London can actually alter the brain structures. I want you to watch the video and see if you can make any connections between what we've been talking about so far and some of the how the structures are influenced by the jobs of the taxi drivers. Definitely an interesting phenomenon. So you'll notice that there's this slide that compares the gray matter between qualified trainees who are taxi drivers and then non-qualified trainees. And just some other details to know that you will notice in the video. So with this study, they had found that there's a connection between spatial navigation in the brain and experience. So basically they found that there's a connection between if the more experience you have as a taxi driver would influence the structure and increase parts of the brain, particularly for this situation, something called the hippocampi. And it just shows that the brain can be changed based on the environment and experiences. And that change is called plasticity. Plasticity is a response to the environment, our knowledge, things that are happening to us. The brain has a lot of plasticity to it. So this study was a real example of that. The next thing I'm going to talk about is the amygdala. The amygdala registers the emotional significance of events and are commonly associated with negative emotions, so fear, anger, things like that. And through those emotions, activate many different regions of the brain. And the amygdala is located just inside the end of the hippocampus. This shows a picture of a rat with no amygdala and a cat. And so we could probably make some predictions about why the rat would still be there and not be scared. Probably because the amygdala is connected to the fear part of the brain. And if you do not have that, then you might not have any experience of fear. So it's a very interesting picture. Okay, let's talk about the basal ganglia. And that's a group of interconnected structures. And evolutionarily is older subcortical part of our motor system. And it connects the motor regions of the cortex with the spinal cord. This structure is necessary for planning and executive movement, and it's critical for initiating and stopping movement in our bodies. And as you can see here, here are different structures of the brain, and the basal ganglia is the red part right here. The next thing would be the thalamus, and it's a subcortical structure. There's two lobes, and it communicates information from the sensory systems and regulates alertness and conscientiousness. And if this region was damaged, it could result in a wide variety of impairments. So just remember that the thalamus acts as a central hub for sensory information, except for smell, and plays a role in regulating alertness and conscientiousness. And just consider the implications of damage to this area. All right, so the hypothalamus is sometimes called the master control of the brain and the body, and that influences hunger, body rhythms, reward-seeking behavior, and then also aggression. It integrates bodily signals with their associated feelings and behaviors. The next is the brainstem, and the brainstem is made up of different parts, including the pons, and the pons controls breathing and related sensations. The medulla oblongata controls autonomic functions, including heart rate, blood pressure, reflexes like coughing and swallowing. So just like automatic functions that we do on a daily basis without thinking about. And then also reticular information runs through the brainstem, and it plays a role in arousal and attention. And when there's different chemical imbalances that could also be associated with ADHD, and it regulates our sleep and wakefulness. So you can see over here the brainstem. Next is the cerebellum, and that supports a variety of functions, including coordination, precision of movement, balance, and then also adjusts our head and our eye movements to help maintain balance. There's parts of our brain that have to do with executive functions, and executive functions are cognitive processes that allow us to plan, focus, focus our attention, and organize multiple tasks and complete goals. That is associated with the prefrontal cortex, and it's kind of like the command center for different processes running throughout our brain, running parallel throughout the brain, and kind of gives us our sense of self. So one case study that heavily influenced our understanding of self and made us think more about the prefrontal brain is from a man named Phineas Gage. And Phineas Gage had been a railroad, part of a railroad excavation crew. And he suffered a horrible accident where an iron rod shot up through his jaw, through his frontal lobe. And what happened is it destroyed the lower and medial frontal lobe called the ventral medial prefrontal cortex. Anyways, that separated a connection between his prefrontal cortex and the rest of his brain. And what happened is a lot of people who knew him really well noticed that he wasn't himself anymore. And so his brain was obviously severely damaged when the rod blasted through his jaw, and it altered his personality and his sense of self until his death. So definitely an interesting story to look into. When we're talking about structures of the brain, it's important to talk about the two different hemispheres of the brain. And as you can see, the brain is symmetrical. And for each structure in one hemisphere, there's a corresponding structure in the other. And a word that was important to know is lateralization. And lateralization means that some functions of the brain are located on either the right or the left side. And some examples would be Broca's area, and that's a region of the left hemisphere that's responsible for language production. And then Wernicke's area, which is a region on the left side that deals with speech and comprehension. The corpus callosum bridges the two halves of the brain. So it's a bridge of fibers that connect the two halves of the brain and allows for rapid communication between the hemispheres and allows each side of our brain to let the other side know what's going on. A term to know would be contralateral, and it's the opposite side organization of the brain. And as you can see here, we have the left hand and we have the right hand, and we have the left hemisphere and the right hemisphere. And one side of the body is connected to and controlled by the opposite brain hemisphere. So we can see here is the left hand will be associated with the right hemisphere, and the left hemisphere of the brain is associated with the right hand. So that's what's called a contralateral control. There's a procedure called split brain procedure and the severing of the corpus callosum to reduce the spread of seizures across brain hemispheres. And one question to ask yourself is like, what do you think the impact of that would be? And as you can see here, this picture, it's another example of how the different parts of our brain are connected. And even the way that we see our right visual field versus our left visual field, those are connected with the opposite part of the brain. So you can see here, the right visual field is connected with the visual area on the left hemisphere. And then the left visual field is associated and connects with the visual right area of the right hemisphere. So I'm wondering if you have any ideas of what would happen if we did the split brain procedure, um, even though we know about that contralateral control. If this procedure is done, one consequence is that information might be presented to each hemisphere independently, but without the corpus callosum, then they're unable to move the information between hemispheres. So the result is the inability to produce information on the right hemisphere with the left hand and the left hemisphere with the right hand. So due to that contralateral wiring, that's not possible. So if there's a separation, the subject might not be able to see something on the opposite visual field. All right, next thing would be mapping the brain, the structure and the function. There's a project called the Connectome Project, and that's just an initiative to map all the neural connections in the brain. And part of this initiative is to define large-scale brain networks and small-scale brain networks. When running experiments in the brain, there is a type of experiment called experimental ablation, and that's removal or destruction of portion of the brain for laboratory animal. And this allows for evaluation of the behavioral effects of damaging in the brain region. So I had mentioned before I'd shown that picture of the rat next to the cat, and how there was no the rat had no response to being by the cat. So they could have performed an experimental ablation with that rat to remove the amygdala or that part of the brain, and as you can see, it's influenced the behavior of the rat. So it says presumably the functions that can no longer be performed are the ones that the region previously controlled. That's the assumption. Another part of experimental procedures would be stereotactic surgery. It's a type of surgery that uses some kind of apparatus to position an electrode on the part of a brain of the experimental animal. And so some requirements of that would be some kind of it's like a device called the stereoaxic atlas or a stereoaxic apparatus. So there could be a radio frequency lesion, and that's a current of very high frequency that destroys neural tissue. There's an exotoxic lesion, which is produced by some kind of cerebral injection of some kind of amino acid, and then a sham lesion, which is like a placebo procedure and duplicates all the steps of producing a brain lesion, except one that actually causes brain damage. Now let's talk about neuroimaging. So there might be some neural imaging that you've heard of just from your experience, from things you hear in the real world. So there's different categories of neural imaging. There's structural and there's functional. So the structural part of neural imaging analyzes the brain anatomy and also can detect and diagnose large-scale abnormalities. When it comes to functional neuroimaging, that analyzes brain processes and detects and diagnoses small-scale abnormalities. This can help examine interface of brain and behavior. So let's start with talking about computerized tomography, also called a CAT or CT. And this computer-assisted X-ray procedure is a series of X-ray beams passed through the head, and it's looking at structure but not the function of the brain. So you can see here some examples. On the left side, it shows just a normal brain, and then on the right side, it shows the victim of a stroke. So the next one would be magnetic resonance imaging, or MRI. And what the construction of an MRI machine would be to look at hydrogen atoms and reaction of the magnetic field. And that gives a good contrast, the differentiation between soft tissues. So that's good for looking at muscles, for the brain, for the heart, etc. So let's talk about the difference between the MRI and the CT. So the MRI and the CT, there's differences between both. Preferred use for MRI would be looking at ligaments and tendons, the spinal cord, if there's a possible brain tumor. Some of the challenges with the MRI would be sometimes it takes longer to perform, it's not as readily available, and it's not allowed. The subject has to be in confined space. For a CT, it's more for people who might have cancer, pneumonia. They're, they could be looking at abnormal chest rays and bleeding in the brain. The next one would be positron emission tomography, or PET scan, and this provides images of brain activity. And it uses some kind of radio-labeled substance, often glucose. And the idea is that glucose will gather at sites with metabolic activity. So the more activity in that part of the brain, that's where the glucose gathers, and that's what it's able to measure. So the positron emission tomography or PET scans compare the regional blood flow between states or conditions, and we can look at different changes over time with a certain condition. For example, experiment can look at the influence of cocaine in brain activity and look at the differences. Comparing the PET and CT scans, they are often performed together, and it can be common for cancer treatments. One continuous full body scan can take about 30 minutes. And the PET scan looks at changes in metabolism versus a CT scan, which can pinpoint exact location and size and shape of a tumor. So the F in fMRI stands for functional, and it measures a change in blood flow related to neural activity. That's often called a hemodynamic response. And the process is called BOLD, blood oxygen level dependence. So you can see on the right side, this shows us the measure and change in blood flow with the neural activity. And it's the cortical activity during hand movement. So what parts of the brain have more activity or more of that hemodynamic response when there's hand movement. So comparing the fMRI to the PET scan, an fMRI has no exposure to radiation. It's better for spatial and temporal resolution, and it's becoming more common, while the PET scan is more specialized. It can obtain anatomical MRI and functional fMRI images within the same session. For a PET scan, it can resolve some areas of the brain better, and these isotopes can be tagged to many possible tracers, so looking at different chemicals like glucose or dopamine, and it can provide different direct measures for metabolic processes. Okay, so let's talk about techniques like transcranial magnetic stimulation, TMS. This would be an example of recording and stimulating neural activities. And this allows scientists to disrupt or enhance brain function temporarily, and that just helps us provide insight into the role of specific brain regions. So these microelectrodes would be inserted in, could be a laboratory animal, and this is like done with a very fine electrode. So there's like some kind of single unit recording that records the electrical activity of a single neuron. And as you can see here for this lab mouse, that electrode is inserted and there's some kind of connecting socket that's there. So this records the electric activity of a single neuron for that laboratory animal. Microelectrodes could be used with human subjects. And it's called electroencephalogram. I didn't do the best job of pronouncing that, or EEG. So that uses macro electrodes placed on the scalp and records electrical brain activity. So the EEG is useful for studying brain waves and diagnosing conditions like epilepsy. So you can see here, these are the different electrical activity patterns that could be seen. So they're used to record electrical activity of a large number of neurons, particularly in particular regions of the brain. And the electrical brain potential is recorded by placing those electrodes on the skull. Transcranial magnetic stimulation of the cerebral cortex is by means of measuring through magnetic fields. And there's different magnetic fields that are produced by passing pulses and electricity through a coil of wire placed next to the skull. And this interferes with different functions in the brain that are stimulated. So you'll see that there's a link here. You can take a look at just to talk about that. And that would be interesting to look at. So deep brain stimulation, there's an example. If you look right here in this link right here, that just talks about like there's different ways to stimulate the brain and the different parts of the brain through different means. So just something to consider and think about for your inquisitive assignment. You may be asked to identify the different kinds of scans that are used to measure function and activity in the brain. And just think about one way to help you remember would be what are the pros and cons of each research method used to investigate the brain. And so remember that there's these different kinds of measurements and strategies like lesion studies, single cell recordings, but you also have the EEG, CT scan, PET scan, MRI, DBS, and TMS. So those are all different kinds of ways to measure brain activity and look at brain structures. And there's kind of like pros and cons of each. So which ones would you say are what are the pros and cons of using each? And then imagine you're recruited as a participant in one of these studies. Which one of these things would you be willing to participate in? Which ones would you not be willing to? So the last thing that I just want to mention would be about neuroplasticity. I talked a little bit about this earlier, and that's the brain's ability to change the structures and the chemicals in the brain to change based on our experience, our knowledge, and just things that are going on in our environment. So neuroplasticity is the brain's capacity to physiologically modify, regenerate, and reinvent itself constantly over the course of a lifetime. And one thing that needs to be studied more and considered is neurodiversity, because even though our structures are similar between all of us as humans, there is quite a bit of neurodiversity when it comes to our brains, and every person's brain is unique. And the wealth of brain connectivity can vary from person to person. So another video that I have linked is a case study of Jody, and Jody had parts of her brain removed. And so it'd be really interesting for you to take a look at that video and to see her experience having that procedure.